So good morning, everyone. Welcome to today's webinar. My name is Lauren. I am the program manager at the 3Rs Collaborative, and I'm really excited to have you joining me today as we have our companies from the 3Rs Collaborative present on their multi-organ microphysiological systems. So I'm just going to start with a quick disclaimer. I'm just going to read right off the slide here. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. So just to begin with a quick introduction on the 3Rs Collaborative and this webinar series. So this is hosted by the 3RC MPS initiative, which has the goal to collaborate to accelerate adoption of MPS in scientific research. And our initiative seeks to increase adoption and regulatory acceptance of MPS in coordination with animal studies. And we're able to do this through our diverse collaborations. So this here is just our board of directors, but our members span across the scientific field. Our MPS initiative includes over 50 different member institutions with over 75 individual members with the companies and institutions shown on the screen here. And our initiative has a few aims: to provide thought leadership and expert consensus from developers combined with other stakeholders, to facilitate appropriate discussion, collaboration, and sharing between developers, and to create direct engagement between developers and regulatory agencies, as well as develop external partnerships and collaborations within the field. And finally, to create resources to facilitate increased engagement with developers. And we accomplish this via our three different work streams: our regulatory work stream, our technology expo, as well as our end user and education. So I just wanted to provide a quick update on what each of these three work streams are currently doing. So we're engaging consistently with end users and providing education. So in 2025, we're hosting quarterly MPS workshops. We've hosted the AI and machine learning, retina and ocular, and this is our our second on multi-organ MPS. And then we have additional webinars for September and December. We also have posters at different conferences, are continuing submissions to Alltech's Corners, and are also looking to organize additional webinars, so stay tuned for that. And you can see any of our previous webinar presentations on our presentations page. Then we also have our regulatory work stream. So in 2025, they've published a publication on perspectives on using MPS for regulatory applications. I put a screenshot of the QR code in the slide there. They're also, again, continuing promotions at different conferences, as well as making forward progress on an exciting cross-platform DILI project. And they're also beginning development of a benchmarking project for MPS end users. And finally, we have our Technology Expo group. We launched our Tech Hub in 2021, and as of 2025, we're continuing to add more disease area and tissue filters, as well as regular promotional posts. And we're also nearly finished an exciting project, which is our Publication Hub. So stay tuned for the launch of that soon. And that's just a screenshot of the hub there. So I also just wanted to highlight that we actually have day 2 of this webinar on Thursday. So definitely make sure you register if you haven't yet. I'll put the link in the chat. And we have 2 additional upcoming webinars for the rest of the year, so make sure you stay tuned for those as well. So for today's workshop, we will be taking questions through the Q&A, so you should be able to access that on Teams there, and they'll be answered at the end of the webinar as time permits. And if you have any additional questions, you're interested in joining the initiative, my email is on the screen there, and definitely feel free to reach out and visit our website to learn more about us. So I'll now hand it over to David from the IQ MPS, who will provide an introduction on them as well.
My name is David, and before we get started with the webinar, just wanted to give a quick overview of the IQ MPS. So the IQ MPS is a collaboration of pharma and biotech companies created as an affiliate within the International Consortium for Innovation and Quality in Pharmaceutical Development. And the IQ has 4 main aims. 1, to develop external partnerships and collaborations to help enhance the inclusion of industry priorities. Also to serve as a thought leader for MPS developers and stakeholder organizations. Also to provide a venue for appropriate cross-pharma collaboration and data sharing, and to create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS in industry. So the IQ is currently composed of 26 member companies with over 150 active participants, which represent experts from drug safety, 3Rs, ADME, and PKPD. And here I'm showing our 2025 Executive Committee. Anya was our previous chair in 2024. And then this year, Rhiannon David is our current chair, Kim Homan is the vice chair, and Philip Hewitt is the vice chair-elect. And on this slide, I'm just giving a breakdown of the IQ MPS structure. The Executive Committee provides leadership and guidance in the management of IQ MPS business and affairs, and helps implement strategic plans recommended by the Steering Committee. And the Steering Committee is made up of 2 representatives from each member company, and they are the primary decision-making body for the IQ. And then within the IQ, we have 4 major work streams, which I'll discuss on the next few slides. And then within each of these work streams, we do have additional several sub-teams. So the first work stream is regulatory engagement. And their mission is to build regulatory confidence in MPS and align on a common understanding of how MPS data should be handled in regulatory submissions by sharing knowledge of MPS utilization and regulators and collaborating to overcome challenges utilizing MPS for drug development applications. Then the second work stream focuses on publications, and their mission is to outline the contemporary pharmaceutical industry perspective and considerations for developing, evaluating, and characterizing MPS models to support drug discovery and development. Then the pilot project workstream works to initiate near-term proof of concept studies for data sharing or prospective collaboration for qualifying MPS systems based on specific contexts of use. And then last, the strategic partnership and communications workstream aims to develop external partnerships and collaborations to help advance inclusion of industry priorities by making the IQ MPS the partner of choice for pharmaceutical MPS education, alignment, and engagement. Additionally, this workstream disseminates IQ MPS positions and learnings and influence external stakeholders. We create a venue for bringing novel information into the affiliate and educate member companies, as well as support MPS adoption among the next generation of scientists. So if you have any questions, please feel free to check out our website or LinkedIn page, or you could contact us directly at info@iqmps.org.
So I'm excited to present today because first I thought, oh yeah, that's easy goal to do multi-organ chips since this is something we focus on already since 15 years. But then I figured out we have so much data, it was really difficult to decide. So I actually added too much data, so please excuse me. So we only have, I will fill up the whole 15 minutes of talk and then be happy to answer any questions tonight or like later this afternoon or this morning in your case, anytime you can send me an email, of course. So what is making TissUse special? So we are really focusing on physiology, like human-like. We connect multiple organs, and some of them are also autologous setup. And for us, it's very important to have the near to physiological circulation system to really allow all the growth factors and cytokines to stay in the system and allow the organs to connect to each other and interconnect with each other. We have the largest organ model and assay portfolio on the market, have a high flexibility to be compatible with different organ models, cell culture types, and inserts, but we are also able to develop personalized chips. We are committed to very high quality standards because I think, as many of us today, we want to pave the way for future assay validation, and we have an automatization to fully automate multi-organ chip assay execution. Our organ models, I think this is very common sense nowadays, is also it can be produced by iPS cells, cell lines, primary cells, biopsies, and precision cut tissue slices. These are some of the organ models that we have experience with. And coming now to the chips, we have developed chip 2, chip 3, and chip 4. The numbers relating to the organ models that can be connected maximum. So each chip 2 can also hold 1 organ model, but it can hold up to 2 organ models. This is a setup of the multi-organ chip. I think many of you have also already seen it. So I'm just running through it. So we have a glass slide on the bottom, just a standard glass slide, then a PDMS layer, which can also be replaced by COC, which is like connecting, then the chip 2 has 2 circulations because each of the chips have the same size. And on the chip 2, we just have enough space so that we had 2 circulation, 2 independent replicates. So we can just focus on one of these here at the side. We have the on-chip pump, which is just pressure and under pressure on a membrane, but with this having a positive flow around. And here at this space, you have a culture compartment 1 and the culture compartment 2. Just like one well of a 96 well, you can include the organ models into this area here and the second in here, and then you have this closed circulation allowing the organ models to interact with each other. And then, of course, you're able, since it's just like one well of a 96 well, you're able to do whatever you can do with one well of a 96 well. You can do 2D culture, but you also can include spheroid cultures, organoids. You can include hydrogels or barrier models in standard cell culture inserts. And of course, at any time point, the whole chip is compatible with live tissue imaging. So this was the chip 296 well, but if you always need to, when you have multi-organs, you always need to downscale at the same ratio. So if you have a liver, like a small liver and a huge intestine, if they are not like downscaled at the same ratio, you will have a problem. So this is why for some of the organ models, we need bigger surface areas, and therefore we developed a chip that combines a bigger surface area with a smaller chip model. And like this, we can still, like the setup is the same as before. Chip 3 connects 3 organ models of these, depending on the size of chips or like organ models that you need. You can decide between chip 3 and chip 3 plus. And then chip 4 is a bit more different. I will go into detail of this chip in a minute. So how do we actually set up our chips? I think all of this has been sometimes heard by some of you as well. So we prepare the chips. We can also use commercial model or biopsy models or tissue slices. Then we prepare them usually in advance and then transfer them into the chip at day 0 and then culture them depending on the different setups for 1 week, 2 weeks, or also 4 weeks. Some of our organ models can also be cultured for 9 weeks or longer and then be ended. During the process or during the experiments, we do sampling. With these samples, we can analyze for metabolite analysis like LCMS. We can look at glucose, lactate, viability like LDH, AST, ALT, albumin production, ELISAs, and of course, if we have floating cells like PMCs in the system, we can also do flow cytometry. At the same time, as I already mentioned, we can non-invasively look at cell morphology and live cell imaging. The nice thing also is that at the end of the culture, we can remove the tissue and analyze the organ models also with histology imaging, qPCR sequencing, do flow cytometry, tear measurement, fluorescein permeation, or also LCMS as an endpoint. These are some of the examples of established multi-organ chip assays that have been published. I think these are 17 publications, but we have now more than 70. So here's another BiomimX assay pipeline. Like I mentioned, we are really trying to push validation also. So we've trying to test all our organ models in different contexts of uses and also in different labs with different compounds. So some of our multi-organ chip combinations coming now to the science. This one is a very old data point, but I really liked it today because I like how you can see how important it is to interconnect the different organ models. So this was functional coupling of human pancreatic islets and liver spheroids on a chip. It was a project with TissUse and AstraZeneca. It started in 2017 and is still ongoing. But the very first experiments, we just used HepaRG cells with stellate cells together. So very simple liver model, connected it with the pancreatic islets of SynVivo primary cells, put them together in the chip for 14 days, and then looked at the glucose consumption and the insulin production. And we can see, if you look at the dotted lines for single cultures here, these two dotted lines, you can see there's hardly any glucose consumption. And at the same time, we have a very high insulin secretion for single culture, quite high islet insulin production, so unphysiologically high. It's nice to have to see this, but in co-culture, you would never have this high insulin production. So what happens now if they're in co-culture? Suddenly, there's a feedback loop of the liver telling the islets to stop producing that much insulin, that they actually have to stop producing it. And at the same time, we also have a glucose consumption, which is much higher than the consumption of both single culture edited to each other. So we can really see that after 48 hours, we reach a physiological level. This, of course, would be reached much earlier if we would have not two organ models, but like 10 or more organ models connected to each other. Of course, now you can say, okay, we produce less insulin because also our islets maybe after 48 hours are not so viable anymore. But we tested this, of course, and we could see that our cells in the insulin produced was stable over the whole time. It was less than in the single cultures at the beginning, but it was stable over the time. And we could actually see that in a co-culture, if we tested for insulin secretion after glucose stimulation, that it was similar to that at day 0, whilst in single culture, we had a slight reduction of insulin secretion in the chip. So the co-culture was actually remained functional for 15 days. We also looked at liver spheroids, but I don't want to go into details here at this point. Next example that I would like to show you is the chip 4, where we connected intestine, liver, kidney, brain, and blood-brain barrier. This was a project with the flagship project of the Horizon 2020, the UTOX RISC, and the HIPSTAR. So we used here the chip 4. So we have 2 layers here. So we have one layer, one circulation is a blood circulation connecting all 4 organ models with each other. Then there's a filtration membrane separating the urine circuit, the excretory circuit from the blood circulation. And here we can see the kidney cells. So we have then the adapter plate, and we have a possibility to add insert-based barrier models here, so that we have a barrier and we can apply substances either orally directly onto our intestine model here, or systemically directly into the blood flow. Then we have the distribution by our circulation, metabolism by the liver, excretion by a proximal tubulous part and a glomerulus part, and we have the possibility for tox tests in an X organoid. In this case, we used brain compartment, but we've also used skin in another experiment, or you could exchange it with cardiomyocytes or whatever you can think of. So in this case here, we've worked with different partners together. So each of them gave us different organ models, and then we just put them together in the chip. So we had commercially primary small intestinal barrier models on transvells from Biotech. We used glomerular cells and proximal tubular cells IPS derived from the BU Amsterdam, Paul Jennings group. We used liver microtissues from SynVivo and neuronal tissues from Biotalentum in Hungary. We then connected them all together and did either oral or systemic application in a co-culture, and then looked at the pathways. So haloperidol was one of the first substances we added. We just put it into the circulation and looked how it behaved. And we could see that the parent compound obviously was directly in the medium. So therefore we could detect it immediately. And then after 24 hours, we could also detect a metabolite here in red. What we could not detect was any excretion of haloperidol. And this is exactly expected because only 1% of haloperidol gets excreted in patients. So we could really simulate the haloperidol excretion as in vivo. Next, we use carbamazepine for oral application, so directly onto the intestine model. We then checked how much of this carbamazepine actually passed through into the blood flow, into the distribution. So we can see here the green is the parent compound, but we can also see very immediately that we have metabolites that are produced and also expressed in the circulation, in the blood flow. In this case, also we can see excretion into the urine circuit, and we can see that it takes a while, and then we can see the parent compound as well as the metabolite compound. One of our customers actually also used this chip and applied an antibiotic, and they applied it at the right concentration, and they could see that they actually had 20% of the excretion into the excretory circuit, which was fine. And just like they expected in vivo. And then they, by mistake, had 1,000-fold higher concentration of this antibiotic. And what they actually saw is that none of this actually stayed in the blood circulating, connecting all organ models, but everything, 100% of this antibiotic was excreted into the kidney circuit. So that was a really nice mistaken discovery by them. So that's showing how well it's actually working. We then also worked on an autologous blood-brain barrier, connecting all organ models derived from one donor in our lab, and then connected it to the blood-brain barrier model where we added endothelial cells on the bottom and the neurospheres here in a transvel, and then co-cultured them. And what we could see is actually, again, we could see this like absorption, distribution, metabolism. And what we could also see here, that depending on the compound, we actually also had penetration into the blood-brain barrier, either in a low amount, a medium amount, or a high amount, depending on the compound. So we have absorption, distribution, metabolism, and excretion in the TIP4.
Ilka, I just wanted to give you about a 1-minute warning for time.
Oh, okay. Okay, I thought I still have. Okay, sorry. So then this is in chip for the immune chip. This was a Gates Foundation Gates Foundation funded chip. So here we connected actually a hydrogel with a chip and then a lung model. And here, which is nice, we also vaccinated the chip first, then infected it with tuberculosis. And this is a completely autologous chip where we have IPS bank for the cells, and then white blood bank for the monocyte-derived dendritic cells, and T and B cells, and then circulating PBMCs in the chip for 14 days. We could see innate cytokine response for primary and booster vaccination, unparalleled T-cell response shown by interferon gamma, and pronounced adaptive CD4 T-cell response. We could also see stable leukocyte clusters. And jumping now, very recent publication, I also wanted to make you aware that we just published our bone marrow model twice, once with TIVR, once with Roche. We've published our kidney injury model, and also a lymphatic vasculature was published by Amsterdam UMC. And with this, I'm sorry that I've run through this so much, but thank you for your attention, and I'm happy to take any questions later.
I'm grateful for the opportunity to talk about how Curi Bio is helping improving the translatability of the in vitro models. And also, I'm super excited to hear about all the great work that our community, our peers in the community are doing, such as Elka TissUse. Today, I'm going to talk about mostly neuromuscular junction platform and neuromuscular junction platform. I will touch on some of the other applications, but if you have any questions about those, feel free to reach out afterwards. But perhaps I can start from an overview, very brief overview about Curi Bio. We're a startup biotech company based out of Seattle, Washington, in the US, and we spun out of the University of Washington. And our main focus excuse me, here we go. Our main focus has always been to deliver human translational functional data to drug developers before they enter those risky clinical trials, basically. We started from 2D platforms, but fairly quickly, based on the feedback we got, we moved towards 3D, especially for cardiac and skeletal muscle applications that were our core focus. And I get to why we did that. But the graph you see on the right is exactly what our focus is to make those in vitro platform engineered muscle tissues behave closer to what we expect them to do in human body. The way we do that is by bringing, I would call, three pillars together. You can have great cell models, you can have great biosystems, and you can have great data analysis, but all of these need to come together and work in harmony. From what we found out, to really move you forward with the model that you're trying to build. And that's exactly what we do at Curi Bio, whether it's our basically product that we're bringing together under that same umbrella of the three pillars, or making sure compatibility with other great products that are out there from one of the providers in our community. And we work across the spectrum from discovery to clinical trials and potency, basically post trials. So if any of those are of interest, let us know and we can help you out. This is the application section I was talking about. We've had a lot of experience throughout the years in cardiovascular, skeletal muscle, metabolic applications. So again, if any of those are of interest, let me know, and I'm glad to connect later to talk more in depth about them. Today, I'm going to mostly focus on the neuromuscular portion and its application in Botox potency. And then talking about those biosystems, we have three main biosystems here. ManArray is designed as a 3D tissue contractility with built-in stimulation. Stingray is our long-term stimulation platform for maturation. And Noli is our optical mapping voltage and calcium system for more mechanistic biology applications. For both 2D, 3D, it's very agnostic to the application. And the biology you have. And then all of these come under the same data analysis umbrella we call Pulse. Earlier I mentioned we started from 2D solution and transition to 3D. Some of these are probably very known at this point to a lot of you, but especially for skeletal and cardiac application, they become more important. I don't have to really touch on cell-cell interaction and that heterogenic kind of like environment that is missing in a lot of 2D applications, but extended culture time and being able to directly measure force are very important for both of those applications. Anyone who's worked with cardiac and skeletal can tell us in great detail that how their experience has gone with, like, at best they're able to keep these cells in 2D for maybe like a week, and after that they delaminate. And that's a problem for some of the gene expression and metabolic changes that that are kind of like metrics of interest for therapeutics. And then the other aspect is being able to directly measure force, which is something that is in 2D is more like a proxy of measurement of the force, whereas in 3D, at least in man-ray platform, you have the ability to actually directly measure how much force they're exerting. And I explain a little bit more on how we do that. So man-ray is a two-post system. Here you see the 3 compartment. Hopefully you see my cursor moving. We have a casting plate at the bottom, a man-ray lattice that holds the 2 posts, and the lid. And the 2 posts, one of them is fixed, which is the black post, and then flexible post there. And basically to cast tissues, you put a mixture of your ECM and cells here in this trough, and then lift the man-ray lattice, move it to a already prepared with fresh media, clear culture plate, and then maintain it there. And anytime you want to do media changes, you can just get a new culture plate, put your media already prepared, you just move all of the tissues at the same time, which also helps with adding drugs or therapeutics. You just prepare that in advance and add all of them at the same time. And the bottom of these plates is clear, as you see here, so you can also do optical imaging with them. And the way we measure force very briefly is that there's a very powerful magnet on the tip of the flexible post. And as the tissue contracts, it pulls on this flexible post and the magnetic field around it changes. And we can measure that and convert it to force with very sensitive sensors. And thanks to all the advancements in the past decade or so, we can just fit many of these sensors underneath the plate and measure force in all of the tissues at the exact same time. And for skeletal muscle applications or some of the cardiac applications, you also need electrical stimulation, which we have this stimulation lid. You can see the top view here that fits here in between and applies stimulation. And you can also apply the stimulation protocol is very flexible, it's well addressable, and you can design up to 24 different protocols if you want, or apply them in any combination. And the software is designed to be very intuitive. You apply your stimulation, and then you go to live view, you see the impact of those stimulations happening. What you can do with these, we talked about longevity in culture. For example, doxorubicin is a cancer drug that has off-target cardiac toxicity. You can see the impact you can test bolus drug dosing and the impact of washout and the number of dosing and those kind of things, because these tissues stay in culture a lot. And you can look at a lot of details when you do that. There's a lot of metrics that automatically gets calculated with their own analytics and report it to you. So it allows you to look at all of the details of what's happening there. The other thing with longevity in culture is that if you have a therapeutics, for example, in this case, we're looking at 5 doses of a drug. You're looking at finding your threshold effect and maximum therapeutic window, or maximum therapeutic effect and therapeutic window. If you look at only first 2 weeks, or let's say first 10 days, and assuming that you get the same improvement in 2D with these muscles, you might assume that the highest dose is doing justice. But as you let it go, you see there's functional toxicity associated with that highest dose that otherwise you'll miss if you can't maintain these tissues in culture longer than possible in 2D. And sorry about that. Very briefly, so we make our skeletal muscles here. You can see those are the ones of primary myoblasts on the left on day 14. Very robust growth for expression, and with iPSC cells, with our Curi iPSC cells, we also see very robust myosin heavy chain response. And then functionally, you can do twitch force in these muscles. You can drive them to tetanus, which is also pretty important. And another very important metric for skeletal muscle is fatigability of these tissues and how much you're improving or how bad the fatigability is. And that's very easily measurable with man-ray. The platform is also the tissues are also compatible with other endpoint analysis. You can do H&E staining. You can look at cross-section, longitudinal, or right down the middle. You can cut the tissues and see how they look. Now, so we have our muscle. now how do we make NMJ? We've been lucky to have a lot of support from NIH and FDA throughout the years, and this project specifically was in close collaboration with both of them. And the goal was to build an NMJ model for screening basically botulinum toxin. In this case, Botox, we're looking at Botox for this specific application I'm showing here. If you look at what is a standard assay for Botox at the moment, it's mouse lethality assay. If you're not familiar with that, it is exactly how it sounds. Basically, you calculate LD50, which is a dose that 50%, half of the mice dies, and then based on that, decide what dose you want to give to humans and release it for other therapeutics applications. And it's also very time consuming, and mice are not human, as we all know. What we did here, so the skeletal muscle portion, we've already covered it. That's the same. What we did is that we have specialized casting plates that we generate neurospheres at the bottom of it. And then we bring these together and we let them mature over the course of 12 to 14 days. And after that, we start looking at if these neurons are initiating muscle contraction with neuromuscular junction. Basically, neurons fire, muscle contracts. And we can look at that because these motor neurons are blue light sensitive. So we also develop our own laser-based illumination lid that basically fits on top of the same plate, and you use the exact same software to drive blue light on that, and then you look at the response in muscle. So example here, you're seeing we hit neurons with blue light, and then immediately right after, muscle is contracting. Now, there are a few cool things here that we can do. Number one, we can see that muscle is responding to neurons. So functionally, there is some connections there. The other thing is that when we electrically stimulate muscles, we're activating all of the muscles, so we see their full potential. When we're stimulating them through neurons, we get a level of force that we can use this as a proxy of innervation, because not all of the muscles are innervated by these motor neurons for sure. And this is something we can use to even further mature the model based on how much the difference is. And when we add Botox as expected, the forces, we get a very nice dose response with doses, of course, and then we start getting responses in muscle, and this is reversible. Once we remove Botox, the response comes back up, and we know nothing's happening to the muscle because we can electrically stimulate it, fine. It's just that whenever we hit neurons, now that Botox is there, there's no response. But we remove Botox, the response comes back. At this point, we've done a number of experiments. This is even a little bit more stale. There's more users, but we've done this with 2 users, 3 different neuronal differentiation, 3 different muscle differentiation. The only things that are the same is instrument and the Botox batch, and we get basically the same EC50 here. The variability is 3%, which the mouse lethality assay that I mentioned earlier has about 20% variability. So it's already a huge improvement there. And then we also started looking at the morphology. We're getting function, but what is happening here? Is there something there? So we look at alpha Bunger toxin presence, which it binds with acetylcholine receptors on postsynaptic membrane of muscle fibers at the NMJ. So if we see presence of alpha Bunger toxin, that also indicates that's a marker for presence of NMJ. We also have some other cool images. I just didn't think I have time to put here some cool images that shows axonal growth from the motor neuron portion towards muscle. But that's available on an application note on our website if you're interested in taking a look at that. And then we also took a look at postsynaptic membrane toxins, which can be used as a key QC metric for your, figuring out when your model is ready and if your model is good enough for your potency assay. Both of those are reversible. Perhaps we see some non-reversible inhibition for acetylcholine mustard, but we just didn't take the experiment long enough to see whether they come back to baseline or not, and that's something that we would like to take a look at more. This is a summary of NMJ. Next step for us is to model diseases here. ALS is one of the diseases that we're actively working, interested, and also collaborating with anyone who's interested in working with those, and hopefully even maturing our NMJ model more. I believe this is all I have. Yes.
All righty. So as you are familiar with Hesperos, was founded by Mike Schuler and I. Mike Schuler is considered the founder of the body on a chip field. And we did 2015. We've been collaborating for at least 20 years. We licensed a bunch of patents from Cornell and UCF, where I'm at, the professor. We've won the Less Prize. We've worked with multiple folks all over the world. We got a 14,000 square foot facility here in Orlando, Florida. We have about 50 people at the company. We're a CRO only. We do not sell our systems. But one thing you might be interested, I'm not going to show this data today, but we were the first microphysiological system using only efficacy data to enable a successful IND that led to the authorization of phase 2 clinical trial for rare disease with Sanofi that has been successful and is now in a 2-arm phase 3. So we're getting regulatory acceptance of these models already for efficacy. So what are we doing differently than most folks? I've been focusing on 30 years this idea of clinically relevant functional readouts. The idea being you go into a doctor's office, he doesn't immediately stick a needle in your arm to be able to pull out biomarkers. He looks how you're walking, how you're talking, he listens to your heart, he checks your reflexes, he might ask about elimination. What he's really checking on is your muscles contracting okay. Is the electric activity of your neurons and cardiac cells working? Is your neuromuscular junction properly functioning? Is the barrier integrity of things like kidney and GI tract working? So I focused my career on integrating silicon-based devices with cells. So like I can put cardiac cells on thin pieces of silicon, contracts, deflects the laser, I can get force, I can pattern cardiomyocytes over microelectrodes, be able to get QT interval, conduction velocity, and beat frequency. Again, I can still get force in skeletal muscle. We have a wide variety of neuronal systems for peripheral nervous system, as well as for the CNS that I won't go into here. And then we can still look at biomarkers or things like liver, but we don't have to. This allows us to look non-invasively for cure, where of course in chronic monitoring these systems, we can take them out to 28 days, but without cell death. We are not using death markers like LDH as our primary readouts. We're really looking at these things that you would have a direct correlation with what people are observing clinically. So what we can do, we can separate, culture out the different cell types on their own chips and their own conditions, and then assemble them to a base platform. We then put a gasket on top of it where we have a fluidic channel that flows over all of the organ systems. And this is all serum-free media. I've banned the use of serum in my lab 25 years ago. And then we have the clear top on it to do all the latest imaging techniques. Additionally, Mike Schuler came up with the idea of the first multi-organ system. I think this is the very first one that he used the equations from a phase 1 physiologically-based pharmacokinetic model to design it. It was published back in 2001 or 2, I think. It's one of our more advanced systems I'll talk a little bit about, where we're doing a 5-organ system. We're doing opioid overdose and recovering naloxone at the same time, looking at off-target toxicity. But the main thing is he's come up with this idea of a pumpless platform. We get rid of all the tubing, all the external or onboard pumps. We get rid of the bubbles, a lot of the complexity. We also make it a low-volume system, so we can see the effect of both parent and metabolite at the same time if we have a liver present. One of the first systems we did with is AstraZeneca, where they had taken a molecule completely through preclinical. As soon as they put it into an animal, they saw a huge QT effect. So they asked us as a validation to do terifenadine, fexofenadine. Terifenadine was an antihistamine that was pulled off the market due to causing long QT. However, metabolite fexofenadine, marketed as Allegra, does not. So see in the cardiac only system, we see a large QT effect. We add in the liver, we see it shift to the right. No effect if the liver is present on conduction velocity, but I see 50 of 33 micromolar with the liver only. And again, we're doing all this without cell death. Cardiac viability is not affected. Now, the nice thing about this is we can actually take the model system, grid it up, and be able to model flow rates, shear stresses. We could also look at absorption and test for it before we add in the cells. So we can actually then look at, say, low absorption versus high absorption. Fexofenadine absorbs a low amount, terfenadine absorbs a lot. We can then look at mixing. We can then look loss due to the absorption, loss due to metabolism, to get the area under the curve. 95% of the literature out there is as dosed, okay, which is very hard to do PK/PD thing, PK/PD predictions without the area under the curve. So we've been able to create these PK/PD relationships where we can account for absorption, pharmacokinetics to that, account for metabolism, pharmacokinetics to that. Again, because we're using these clinically relevant functional readouts, we can actually create the pharmacodynamics that one would expect to see in vivo. In this case, terfenadine acts on the hERG channel inside the cell, so we can actually look at the bioaccumulation inside to be able to get the pharmacodynamics of how terfenadine works. We're able to use that data and compare it for calculating in silico, okay, the data they had for guinea pig, dog, and non-human primate. This was published with AstraZeneca Nature Scientific Reports in 2019. Now, Roche said, oh, that's a very nice toxicity model. Can you actually look at efficacy and tox in the same system? So what we did, we took the heart liver system, they added in a multi-drug resistant cancer and a non-multi-drug resistant cancer. And what we did is we then looked at the tamoxifen by itself is not a good chemotherapeutic, but for the non-multi-drug resistant cancer, we see that we add in the liver, we do see the effect. We add in metabolite directly, we see the same effect. For the multi-drug resistant cancer, you basically see that for tamoxifen, no effect, with the liver, no effect, with its metabolite, no effect. But we add in a drug, verapamil, which blocks the PGP channel, which is basically pumping out the chemotherapeutic as fast as it goes in. We do see a decrease in proliferation, no additional decrease in proliferation. So verapamil is not acting as a chemotherapeutic. Now, the really interesting data was that if we took tamoxifen by itself, we saw a decrease in conduction velocity but recovery, decrease in force but recovery, small decrease in beat frequency but recovery. However, for the drug-drug combination, you see for conduction velocity, it goes down but does not recovery. Force goes down but not recovers. But now beat frequency, if you were just looking at beat frequency as your readout, you would almost say, okay, this is acceptable toxicity probably. But actually, when you're looking at the functional readouts, that is not the case. So again, this is really important for oncology, where many of the therapeutic applications anymore are drug-drug combinations. People have a pretty good idea how the efficacy combines. In many cases, no idea how the off-target toxicity combines. And again, this is no effect is cell viability. It is published in Science Translational Medicine with Roche back in 2019. We can also create comorbidities in these models. We've created sarcopenic, cardiomyopathy, and MAF-LD models. We're doing combinations of those, okay? Again, we can take these things out to 14 days and maintain the phenotypes, okay, for all of them. And the first publication for the combination of sarcopenia and cardiomyopathy with normal liver, okay, is in preparation. We also had done a work with Vanna Pharmaceuticals, had been asked by the FDA to do a 9-month Beagle study for their compound. What they did is they did a 3-month Beagle study and approached us. Could we do a 4-organ model system and then use that with a 3-month Beagle study to replace the 9-month Beagle study? And that was successful. They were actually allowed to go into the IND review. So again, what we did is we can actually take out liver in this case, cardiac cantilevers, cardiac for conduction velocity, neuronal models where we can actually do LTP with the models, and also kidney in the same thing, where we did 2 controls for each organ system. and it monitored these clinically relevant functional readouts in this multi-organ system. I'm only going to focus on a couple. Again, we can look at biomarkers. Again, in the system, we look at troponin, the terfenadine, doxorubicin causes deficits. However, we were also looking at low, medium, and metabolizing liver, and showing that there was some effect, okay, at the lower concentrations, having to do with the different metabolizing levels of the liver. You see, again, even at the super physiological concentrations, we see the effect of the biomarker. And then you can see how for cardiac force, you do see the two controls go down, whereas their molecule is fairly stable. For beat frequency, you go down and see it go down very quickly for the two controls. Okay, very safe levels of toxicity. Okay, for conduction velocity, you see the QT interval. Doxorubicin, known to be very toxic, goes down and does not come up. Terfenadine ends up going down with time. Again, same thing is maintained as we have higher and higher concentration. We could also look at cortical neurons. Again, learn long-term potentiation assay, see that there was not a lot of toxicity in the neuronal systems, kyanic acid, norepinephrine, okay? But we do see at the highest level, we do see a stratification between low, medium, and high, suggesting that the parent has some neuronal toxicity at supraphysiological concentrations. And then lastly, looking at KIM1 levels, you do see gentamicin, in case it kills kidney cells very quickly, as maintained at all concentrations. And we do see that gentamicin causes KIM levels to go up, okay? And at the highest concentration, you do see a little bit of kidney toxicity. Again, this was the data that was included with that 3-month Beagle study to avoid doing a 9-month Beagle study. We can also take a look at these systems and looking at opioid overdose and recovery, at the same time looking at off-target toxicity. The first thing we had to do with the model is differentiate out pre-BOTC neurons, which are the neurons that control rhythmic breathing. We were able to do dose response curves, okay, for 4 different opioids. And then the key experiment was before opioid addition, we were able to see nice electroactivity. Opioids shut it off, but naloxone turned it back on. This was published in Advanced Biology in 2023. Now, we wanted to do was to now look at this in a multi-organ system, look at off-target tox. We could take out all these systems out to 28 days. Okay, and clearly see the important slide here is you have the controls, fentanyl takes everything down, but with repeat dosing, we could basically see the recovery, see the fentanyl go down, recovery naloxone, fentanyl go down, recovery naloxone, fentanyl go down, but recovery with naloxone. Now, what was interesting is we also saw for contractile force, we did see at the highest time frame, we did see an effect. Force went up in naloxone, but it tended to balance each other out. However, for beat frequency, we saw with fentanyl go down, naloxone initially go down, but stabilize, but no recovery with naloxone, which suggests this is one of the reasons why fentanyl is so hard to reverse. And this initial acute work was published in 2024. We also, with the Gates Foundation, asked us to create a malaria on a chip model. We used splenocytes, liver, and endothelial cells. Again, we created a multi-organ system where you can actually add blood to it, reproduce the entire malaria life cycle for the blood, and we can monitor all these different parameters and be able to determine the active concentration. And for chloroquine, which is a widely used anti-malarial, we're able to get the PK profiles for different dosing, the PD profiles. Okay, again, you saw the control. You see that for medium concentrations of chloroquine, you see the recrudescence. We're able to calculate the maximum tolerated dose, no observable effect level dose, the EC50 in our model, which correlated very well with the in vivo uncomplicated malaria. And we can then put that data into an in silico model, okay? And we had done an acute dose with it. We can then use the in silico model to be able to create the in vivo pharmacokinetics, to be able to use the in vitro data to predict the in vivo pharmacodynamics and get very close to what they see after chloroquine treatment.
James,
Lastly,
just a 1 minute time warning.
2 slides left. We also can create skin models, kidney models, as I showed, BBB, GI tract. We've been able to put these together to be able to look at oral versus intrathecal versus IV dosing. And then we've also, these are the clinical trials and the support of our human tip data, basically where the majority of them were repurposed drugs using only our efficacy data. Again, these are all the names, okay, and the clinical trial numbers for that. And lastly, again, our business model is to custom build what folks want. So we can do very mix and match all kinds of different combinations in these multi-organ systems with the functional readouts. Thank you very much for your attention.
All right. Just a second. Oh, for some reason it went out, sorry.
Yep, that looks good.
All right. Thanks a lot, Lauren, for introducing me. And thank you everyone for attending this webinar today. I will be presenting some data on our new model system in which we developed a multi-organ on-chip model system for studying the lung-brain axis, in which we studied the effect of inflammation on leukocyte dynamics and the inflammatory response in it. Now, coming to the lung-brain axis, the COVID-19 pandemic really showed us why it is important to have these in vitro models, and specifically looking at the way the pathophysiology of the infection. It is very critical to have a vascularized component when we are modeling these systems. So just to give a brief information about the COVID-19 infection, when the viral infection is present in the lung, it produces pro-inflammatory cytokines such as TNF-alpha, IL-6, and IL-8. And we also see production of chemokines such as CCL2, which are responsible for attracting multiple immune cells to the site of inflammation. Now, this leads to the production of pro-inflammatory mediators and ultimately the cytokine storm, as we know, which then spills into the blood vessels and ultimately goes into the systemic circulation. And also through this axis, the bloodstream, it reaches the brain, where it can disrupt the blood-brain barrier and also cause neuroinflammation over there. And the brain tissue in itself starts producing anti-inflammatory compounds such as TGF-beta and IL-10 in order to counteract the inflammation coming from the lung. So the vascularized component plays a critical role in this axis in causing the neuroinflammation which is coming from the lung. Now, in order to model this, the SynVivo platform plays a critical role because our platforms have unique features which help us in modeling this system. One of the important features of our system is the side-by-side architecture, which helps us in visualization of the cells in the lumen as well as the tissue at the same time. So that's an important feature where you can see the cell-cell interactions. Now, additionally, the microvascular lumens also facilitate the integration of immune component. You can see the circulating immune component in our models. You can visualize them and have real-time visualization of the immune response. Also, the model overall helps integrate the blood flow, the immune component, and the 3D tissue architecture. So it clearly mimics the in vivo-like conditions. And which then enables the introduction of infectious agents, drugs, and therapeutic candidates in a controlled and realistic environment. Our inflammation model is highly adaptable and has been successfully integrated with other organ-specific models, demonstrating its broad applicability and potential for translational research. Now, uh, to give a bit more about the model helps us in creating, if you see over here, a vascularized vascularized component in which we have a vascularized lumen which forms after seeding, which is clearly mimics the blood vessels, the in vivo like conditions, and also we also see tissues can be created which are integrated in the model along with the lumen and thereby mimicking the actual fluid flow component to the tissues. Now, as shown to the right, our model also helps in mimicking the architecture of the blood vessels. This is particularly important because the architecture is the bifurcations which you see in the architecture. They are critical to replicate the branching uh, in the blood vessels, and the immune cells, precisely the dynamics of the immune cells are different in this architecture because this affects the cell adhesion, trafficking, and also the signaling dynamics when the immune cells are flown through this. So this fidelity is crucial for understanding the pathophysiological processes in situations such as the cytokine storm, which is seen in COVID-19 and its impact on multiple organs. Now, in terms of modeling this system, we have developed a lung and brain axis model in which we have the lung component, as you can see over here, and it is connected through a vasculature to the brain tissue. Our brain tissue contains the astrocytes and pericytes in the tissue compartment and brain-specific endothelial cells in the vascular component. And then the connection vasculature contains the endothelial cells, UVEX, followed by the lung vasculature, which contains UVEX cells, and the lung epithelium, which represents the lung tissue. Now, before moving to the whole component of multi-organ, I just want to say in our previous work, we have modeled the neuroinflammation in our BBB model, in which we used compounds such as TNF-alpha to induce neuroinflammation. And at the same time, we also introduced anti-inflammatory agents which targeted PKC delta. Now, in doing so, we studied the effect of these inflammatory agents and anti-inflammatory agents on the BBB integrity, as well as the effect of this inflammation on leukocyte adhesion. We were able to model this, and as you can see over here, once the TNF-alpha is introduced in that group, we can see disruption of the blood-brain barrier. This was studied using a 4 kilodalton FTSE-dextran, and we see the permeability increase. This is due to the disruption of the blood-brain barrier. Whereas when we introduce an anti-inflammatory agent, we are able to counteract the inflammation, and it is almost as same to the levels as same as the vehicle control. Now, in terms of the leukocyte adhesion, we see over here, you can see over here the immune cells. These were primary neutrophils which were introduced in an inflamed vascularized component. And you can see over here the adhesion of the neutrophils in the group in which TNF-alpha is introduced. And we see a reduced adhesion of these neutrophils when an anti-inflammatory agent is added. So this clearly helps us understand that our models are able to mimic the inflammatory cascade. And then now the important component of any inflammatory model, as we know, is having the whole blood component would be great. So in this case, to model that, we used 20% volume by volume whole blood, which was created by diluting whole blood from the donor by using Hank's balanced solution to 20% in order to dilute the hematocrit. And then we stained the PBMCs and reintroduced them in the blood so that we are able to visualize them. Now, once created, these PBMCs and the blood were introduced in an inflamed vessel. The vessel was inflamed using TNF alpha, and the PBMCs were flowed for 10 minutes. And as you can observe over here, uh, we are able to see attached leukocytes which can be counted, and this can give us an idea that we are able to model the uh, inflammation in the blood component. Now coming to the multi-organ system, the first step in establishing this is to establish the individual tissue structures. We first establish the vascularized component in both the tissues, and then the individual lung tissue and the brain tissues, the astrocytes and pericytes, are seeded. And once the whole multi-organ component is established, then we incubated the lung tissue specifically with a cytokine cocktail, which is a pro-inflammatory cytomix of TNF alpha, IL-1 beta, and interferon gamma, which causes the inflammation in the lung compartment. And then after 4 hours of this incubation, we rinse the lung tissue and fresh media is flown through it. Now, when we flow the fresh media, the compartment of lung and brain is connected, and the media is flowing from the lung to the brain to mimic the inflammation which will happen in a pathophysiological condition. Now, we flow the media for 1 hour across these tissues, and then in two separate assays to study this inflammation, we introduced PBMCs, the stained PBMCs in this model, and after flowing them for 1 hour, we count the adherent PBMCs in the inflamed vessels. And in a separate assay, we collected effluents at various time points to see the cytokine quantitation and to see if there was any effect coming from the lung inflammation to the brain tissues. Now, we saw interestingly that the number of adherent PBMCs in the cytomix treated as expected were present in the lung vessels. We see significantly higher number of PBMCs adherent in the vasculature component of lung, but also we saw that translating to the brain vascularized component, where we saw a significantly higher number of PBMCs attaching to the inflamed vasculature in the brain, which is connecting from, which is coming from the inflamed lung mediators, the inflammatory mediators coming from the lung compartment. In addition to this, we also saw an increase in CCL2, the chemokine which I mentioned previously, responsible for attracting immune cells. We saw that at 3-hour time point, it was significantly higher in the brain compartment, thus successfully modeling the lung-brain inflammatory cascade similar to the COVID-19 response. And in addition to this, we also saw an increase in IL-8 at 3 hours. And since this was not present in the cytomix cocktail, this is definitely a tissue-driven cytokine production coming in the brain as an inflammatory response. In addition to this, we also saw an anti-inflammatory response coming from the brain, IL-10, which is an anti-inflammatory agent. We saw its production being significantly higher in the brain vascular component, thus successfully modeling the whole lung-brain inflammation cascade, and particularly the neuroinflammation cascade, because these inflammatory responses might be coming from the brain tissue as well as the endothelial compartment of the brain.
Andreas, I just wanted to give you about a 1-minute time warning.
Sure. So to summarize, we were able to, using the multi-organ system, we were able to study the circulating immune cell dynamics. As you saw, the addition of PBMCs was one thing which showed that in the and also we were able to see the inflammatory responses coming particularly from the brain using the BiomimX data. So overall, the SynVivo vascularized interconnected models, they enable to model the immune-mediated inflammatory response in a realistic manner. And this is critical for developing therapeutics that maintain efficacy while reducing the off-target effects. And you will be able to study if there are any off-target effects of your compounds on our brain tissue. And we offer these models as products, as well as we offer our services to study your compounds. So please contact us if you want to know more about our models.
Okay. So thank you for introducing me, and thank everyone for attending this webinar. I'm Erika Ferrari. I'm a commercial science leader in BiomimX, and today I will present our multi-organ on chip platform for the in vitro investigation of off-target cardiotoxicity and synovium cartilage crosstalk. Okay. So as everyone here knows, we work in the field of organs on chip and multi-organs on chip in drug development, and in particular, these models have high capabilities for vector generating a precise model of a human-based organ with increased success rate of the drug development, as well as reducing the cost related to drugs R&D. And in particular, in BiomimX, what we do is that we develop beating organs on chip, meaning that we are building on 3 key components. So the use of human cells, we embed them in a hydrogel, so to provide a 3D microarchitecture, and then we provide also mechanical forces. And these mechanical forces have been applied to the tissue through the UBIT technology that is able to precisely stimulate the tissue so that the cells would experience a movement which is similar to what they would feel in our body. And by really tailoring the mechanical stimulation, we can generate either through human models, so help and enhance the differentiation of maturation of human tissue in vitro, but we can also modulate the mechanical stimulation to generate models of complex diseases. And this is very important, in particular in this moment, because also regulatory bodies are moving towards this, the adoption of this alternative to animal models, and we are very active in this field. For instance, our president is also president of the European Organs on Chip Society, and we are actively discussing with the European Medicine Agency to regulate our cardiotoxicity assay. And in particular, our technology has been applied in different applications. So we are working a lot in cardiac related applications, both physiologic and pathologic, cartilage, osteoarthritis, also barriers like applications such as the lung and the gut. And in particular, for today, I will show you our multi-organ on chip models in the field of cardiac and osteoarthritis application. I will start with our multi-organ on chip for safety, and in particular here we develop a model for the in vitro investigation of off-target cardiotoxicity of liver metabolized drugs. And this is our device. So it's a PDMS microfluidic device. This is the chamber layer where we host the cells. And in particular, we can host the liver cells in the liver chamber and the 3D heart cardiac model within the cardiac chamber. On top of it, we have a microfluidic valve system that precisely controls the communication between the two compartments. Indeed, this is a normally closed valve system that can be opened when a negative pressure is applied. And on top of it, we have the actuation chamber, which is meant for the application of the mechanical stimulation that is very important to mimicking the heartbeat for cardiac cells. And to give some details about the models themselves. So the liver model is based on the micropattern co-culture model, where we have FG2 island, so islands of hepatocytes surrounded by fibroblasts. And this model has a good viability and good albumin production, and has been already validated for metabolism of anti-cancer drugs such as Tegafur. While our cardiac model is called the UART and is composed by 75% of cardiomyocyte. We use both IPS or primary cells and 25% of human fibroblast. And these cells are mixed in a fibrin hydrogel, which is then injected in the channel of the device. And after 1 week of mechanical stimulation at 1 Hertz, we see that the cells appear very interconnected with the striation of the sarcomere. We can also see that the cells really beat all together like a syncytium. We also can control and monitor their electrical parameters, and the mechanical stimulation also enhance the ion channel expression. So what we did was to combine these two models within our UART and liver platform, and we used it to investigate the effect of terfenadine after liver metabolism. Indeed, terfenadine is a cardiotoxic drug known to prolong the QT interval of the cells. And what we did here was to first seed the cells, so generate our liver and cardiac model within the platform. Then we administered the drug in the liver chamber. We let it metabolize for 1 day. Then we opened the valve system and we allow the metabolites diffuse toward the heart compartment, and then we can perform the analysis. Firstly, we wanted to be sure that the liver model was able to precisely metabolize terfenadine. So we ran a mass spectrometry analysis, and here we can see that indeed the liver is able to correctly metabolize the compound. Indeed, we see a peak of fexofenadine at 2 minutes of mass spectrometry. From the viability point of view, we first checked the viability of the cardiomyocyte, and as expected, we can see that when terfenadine is directly administered on the heart cells, we see a decrease in viability of the cells, while this is not happening when it is firstly metabolized by the liver, following the trend that is more similar to the control. And we also checked the viability of our liver model, and both the control and the drug administered the liver had a good viability. Then we also checked the functionality of our cardiac cells by means of field potential recording. And again, here, as expected, we see an increase of the field potential duration when terfenadine is directly administered on the heart cells, while this is not happening again when it is firstly metabolized by the liver. So our liver heart models allow the testing of non-cardiotoxic metabolites generated from a cardiotoxic drug. And to conclude this first part of the presentation, we can see that by integrating a liver compartment in our UART model, we see a key role of the liver metabolism in drug toxicity studies, here presented with the case terfenadine, and also that liver micropattern co-culture can metabolize cardiotoxic drugs before they are administered to the heart, so better recapitulating systemic toxicity. So not only a relevant microenvironment, but also multi-tissue complexity is required to recapitulate liver-mediated cardiotoxicity. Of course, this model has a limitation. Indeed, the liver model is a 2D liver model. So what we have been doing in BiomimX is to build on this liver heart model to develop a bicompartment 3-dimensional multi-organ on chip to study tissue crosstalk. And this new advancement has been used by us in efficacy screening, and in particular we developed a model for the in vitro investigation of synovium cartilage crosstalk. And this platform is very similar to the one that I presented previously, but here we have both compartments that are 3-dimensional. And indeed, we can see the synovial compartment here in blue, which is, let's say, static 3D microtissue composed by synoviocyte and macrophages. In green, we see the cartilage model that is indeed compressed. So here we are applying the UBIT technology. And we have as well, again, the valve system that allows the confinement, but also connection of the two compartments when needed. And just to give some details about the two models, so the cartilage compartment is based on BiomimX uni model, which is the first human model of human osteoarthritis, and how we generate it. So we usually start from human healthy chondrocyte, we put them in a hydrogel and within the platform, and we culture it for 2 weeks to achieve native-like cartilage. And after this, we use our mechanical stimulation to induce osteoarthritis traits. Indeed, we see an increase of degradation, as you can see from the increase of MMP13, an increase of inflammation, as you can see from IL-6, and also the acquisition of hypertrophic traits. While the synovial compartment, we call it USYN, and we generate it by starting from the injection of synoviocytes and macrophages within the synovial compartment, and then we apply a pro-inflammatory stimulus made of TNF-alpha and interferon gamma, and we apply it for 3 days. And after 3 days, we remove the stimulus and we check what's happening at the system at day 7, because we want to make sure that the inflammation is maintained, but without having the stimulus itself still within the system. And as you can see here, at day 3, indeed, we see a higher expression of inflammation and degradation, but this is still a bit maintained at day 7, which is what we wanted to achieve. And from the macrophages point of view, here again we see an activation which is higher at day 3, but this is still maintained at day 7 of culture. So we asked ourselves what would happen if we inflame one compartment, then we open the valve and see the effect of the inflammation in the first compartment towards the second compartment. So what we did here was starting by inflaming the synovial compartment in the way that I just showed. So we inflame the synovium, and we have a healthy cartilage here. So after the synovium has been inflamed, we open the valve and we see what's happening on the healthy cartilage tissue from like stimulus coming from an inflamed synovium. And here we can see that in the cartilage, we see an increase of inflammation like IL-6 and an increase of hypertrophy such as IL-10 and a decrease of collagen 2 and aggrecan. So meaning that the inflamed synovium has a key role in triggering also cartilage degradation and inflammation. So we did the opposite. So here we started by altering the mechanical environment of the healthy cartilage tissue, so to induce the osteoarthritic trace thanks to the UV technology. So we apply our hyperphysiological compression and we have a healthy synovium. And after we developed the OA model, we open the valve and we see what's happening on the healthy synovium on the other side. And here, surprisingly, what we see an initial protecting effect of the synovium upon cartilage mechanical damage. Indeed, we see a decrease of IL-8 and an increase of lubricin and collagen 1, but we still see macrophagic activation, so meaning that there is still an early OA onset. So to conclude the year, by integrating synovial compartment in uni, we can see a key role of synovial inflammation in triggering cartilage degradation and also an initial protective effect of the synovial membrane on the cartilage model. And what we are doing here is to have a more comprehensive drug efficacy assessment capable of distinguishing the contribution of different cell components on the uni and UCIN model. So not only, again, here a mechanical relevant environment, but also multi-tissue complexity is required to recapitulate OA. So to conclude, this is what we are doing in BiomimX, so to develop advanced culture tools that can be used for target identification, lead optimization within safety and efficacy application. And our data are used for R&D application and to generate human-centric models. And with this, I would like to thank my group and all our collaborators, and thank you all for your attention.
Okay, fantastic. Hi, everyone. Well, thank you so much for sticking around to the very end of this webinar. I'm really excited to be with you today. My name is Lizzie. I serve as AIM's Biotech's business development manager and technical lead based out of Boston. And today I'm going to discuss our humanized 3D blood-brain barrier model that we use for oncology and neurodegenerative disease drug development. And actually, this is a picture of our blood-brain barrier, which is just a really pretty image in my mind. Okay, so just as a quick overview, I want to mention that AIM is an MPS startup that came out of Roger Kam's lab at MIT. And our mission is to make human data more accessible throughout all phases of drug development. We achieve this by enabling researchers to create biological structures like perfusible blood vessel networks, mimic disease processes such as disrupted BBB transport in CNS disorders, investigate mechanisms of action of cell-based immunotherapies to target cancer, and screen candidate compounds under more human-relevant conditions such as dynamic and perfusible tissue microenvironments that are achieved through vascularized organoids. This deep biology is created in simple 3-channel microfluidic devices called identics that are standardized, automation compatible, and scalable up to 40 experiments in SBS-compliant microplates. Because of our design, no external equipment like plate rockers or pump systems are required to achieve perfusion. Additionally, our newest model, OrganMPS, will be highlighted in today's talks as it enables larger tissue samples up to 2 millimeters in diameter to be cultured within these 3D tissue microenvironments. So although we have all those different applications, I'm going to focus our talk on one of the most important organs of our body, which is our brain, and specifically the way our technology enables users to co-culture the BBB with different cellular entities to form complex biology and investigate therapeutic modalities to treat various diseases. As we're all well aware, the BBB is critical to maintaining brain homeostasis by controlling the transport of compounds in and out of the brain. The specific interactions and spatial organization of the 3 major cell types, so the Brain endothelial cells, the pericytes, and the astrocytes that make up the BBB are responsible for supporting the barrier function. Dysfunction of the BBB is implicated in aging and various diseases such as Alzheimer's disease, Parkinson's disease, and brain tumors. Traditionally, BBB studies are conducted either in vivo using rat models or in vitro using transwell assays. The in vivo models are limited by species differences and challenges to investigating mechanisms of action, as well as cost, time, and ethical considerations. On the other hand, transwell models do not allow for the proper cellular interactions and spatial organization of BBB cell types, leading to non-physiological measurements of BBB transport and limitations on investigating BBB-specific active transport. So to address the limitations of those existing models, AIM developed a human-relevant 3D BBB model that uses the self-assembly of the three BBB cell types in a 3D hydrogel to form perfusible vascular networks over the course of 7 days. The process is called vasculogenesis, and it allows for the physiological formation of relevant cell-to-cell and cell-to-ECM interactions that are critical for maintaining BBB integrity. To validate the spatial organization of the cells, we use IF staining to measure CD31 expression to identify the endothelial cells, shown in green, phalloidin to identify the pericytes, shown in orange, and GFAP to identify the astrocytes, shown in red. As you can see in these representative images, as well as this movie, the pericytes are able to wrap the endothelial cells, and the astrocytes are able to form end feet in contact with those endothelial cells to reinforce the BBB. And again, these cell-to-cell interactions, which are really critical, cannot be achieved in a transwell, and animal models lack the human relevance and ability to actually visualize these interactions. To assess barrier function, perfusion of fluorescently tagged molecules of different sizes can be added to the model, and images of the diffusion of the compounds into the tissue can be captured over time to quantify the permeability. We see that smaller compounds are able to readily cross the BBB, whereas the larger molecules, such as human IgG, are unable to pass through, and the tightness of the BBB, as measured by permeability, is on the order of magnitude of the in vivo measurements from rat models. In contrast to TIR measurements that only measure ion transport and do not measure active transport using BBB-specific transporters, we confirmed our model enables selective and targeted transport of antibodies that bind to transferrin receptor to cross the BBB. Our results indicate that MEM189, which is a monoclonal antibody that specifically binds to transferrin receptor, can readily cross the in vitro BBB barrier more readily as compared to the larger dextran and human IgG, indicating a selective and functional barrier. To mimic dysfunction of the BBB under disease conditions, we have applied a cytokine insult and showed that treatment of the BBB with inflammatory cytokines such as TNF-alpha and IL-1 beta leads to dose-dependent disruption of the BBB as measured by increasing permeability of fluorescently tagged dextran. With the removal of TNF-alpha treatment, the BBB is able to recover after 24 hours. Additionally, the combination of TNF-alpha with IL-1 beta induces BBB leakage at a lower dose as compared to the cytokines on their own. And finally, the impact of oxidative stress, which can occur under disease states, was measured through the addition of hydrogen peroxide, which similarly showed a dose-dependent increase in permeability. So taken together, these findings validate the phenotype and function of our BBB model. And now I want to share a few applications where we combine the BBB with other cellular entities to form disease models for drug development. So the most common adult primary malignant brain cancer is glioblastoma, which has very dismal patient prognosis, and novel treatments are desperately needed to prolong their survival. A common challenge in treating GBM is drug transport across the BBB and avoiding treatment resistance. And so using our larger tissue sample platform, the organics model, we're able to culture tumor spheroids within a BBB network to investigate drug development and treatment of GBM. So in this work, a PDMS-based precursor was used to validate the experimental approach, which consisted of culturing U87 spheroids within the BBB, and IF staining was used to confirm the spatial arrangement and phenotype of both the tumor as well as the BBB. Permeability of different size compounds was evaluated with and without the tumor spheroids being present, and it indicated a higher permeability for lower molecular weight dextran and increased variability in permeability for BBB networks cultured with the U87 spheroids. And importantly, all of these values were on the order of the in vivo rat brain permeability. And finally, the tumor growth was quantified for U87 spheroids cultured with and without the BBB, and sensitivity to the first-in-line chemotherapeutic TMZ was shown to be reduced when the U87 spheroids were co-cultured with the BBB, suggesting that increased spatial organization of the U87 cells within more physiologically relevant microenvironment affects TMZ sensitivity. Beyond chemotherapeutic agents, our BB model can also be used to investigate efficacy of novel CAR T cell therapies targeting GBM spheroids. So in this work, the organics platform was used, and we again cultured the U87 spheroids within a BBB network. And after 7 days, candidate CAR T cell therapies were perfused through the vasculature. Through quantification of the tumor area, we see that the untreated samples had the greatest increase in tumor area, whereas when you add the CAR T cell therapies, that tumor growth was reduced. And then, although not shown here, data from in vivo xenograft models confirm the findings from this in vitro tumor BBB model, which validates the utility of our platform in enabling the testing of CAR T cell targeting and extravasation, with the presence of astrocytes and periocytes, allowing for the observation of off-target cytotoxicity, providing a more accurate assay of CAR T cell function in vitro. And finally, to demonstrate the targeted delivery of drug-loaded nanoparticles to treat GBM, researchers from our co-founder's lab, Roger Cam, used a modified PDMS version of our IdenTix platform to culture patient-derived xenograft GBM cells within the BBB network. Through quantification of cytox signal, which indicates disrupted cell membranes, their team saw no differences in signal near or far from the tumoroid when comparing free cisplatin-loaded nanoparticles and nanoparticles that are specifically targeted towards angiopep2, which binds LRP1 transporter on BBB cells. Within the tumor, however, the specific targeting of AP2 cisplatin-loaded nanoparticles, in fact, did induce the greatest cell death, as indicated by the highest cytox signal. And so overall, these findings demonstrate that our BBB model can be combined with various cellular entities to form oncology disease models and evaluate novel therapeutic modalities. And as a final application, I want to highlight that in addition to forming these oncology disease models, our BBB model can also be applied to study neurodegenerative diseases such as Alzheimer's and Parkinson's disease. Again, using a PDMS-based version of our organics model, our co-founder's lab, Roger Cam, demonstrated co-culture of the BBB with brain organoids harboring single-point mutation disease-related point mutations that correlate with the disease phenotypes. Using measurements of permeability of two differently sized dextran molecules, the researchers showed that co-culture with the diseased organoids, shown in blue here, induced leakier BBBs compared to co-culture with non-disease organoids, shown in purple, for both Alzheimer's as well as Parkinson's disease. This work highlights the ability of our platforms to enable development of human-relevant models for studying disease progression and disrupted BBB transport that are characteristic of different CNS disorders. And so continuing to develop and characterize these disease models is an active area underway at AIM Biotech, as well as the development of an isogenic iPS-derived BBB model to improve the reproducibility and standardization of this model. We're also looking to incorporate the immune system by culturing the microglia within the brain tissue, as well as perfusing circulating immune cells through the vasculature. And finally, we are continuing to evaluate targeted drug delivery using receptor-mediated transcytosis to improve drug delivery and evaluate BBB transport for novel therapeutics. So if you would like to work with us, please reach out. We offer our IdenTix and organics platforms as consumables, and we offer contract research services to support your drug development, drug screening, and custom model development. And then in an effort to help streamline experimental workflows and again, improve reproducibility of vascular MPS models, we recently launched our cell system products, AngioPrime and VasQKit. AngioPrime is an assay-ready angiogenesis product featuring preloaded IdenTix 40 plates used to screen angiogenesis-related compounds. And the VasQKit provides users with our MPS platform, the cell types, and the ECM that are required to form perfusible blood vessel networks to create disease models and evaluate drug transport across the vasculature. And so I just want to say thank you for your time, and I'm happy to answer any questions. Please feel free to reach out to us. My email is shown here. Also follow us on LinkedIn if you want to stay up to date on our latest announcements. And if you'll be attending the MPS World Summit in a few weeks, we will be there. So please drop by our booth 409. And if you'd like to attend our educational workshop, we can also give you some hands-on training using our platforms. So thank you so much, and I'll be happy to take any questions.
Amazing. Thank you so much, Elizabeth. So if all of our presenters can come on screen now, turn on your camera and your mic, and we have a few questions ready. So I'll go through those. And I think we've got nearly everyone here. So I'll start with the first question, which is kind of a more general question for all. In terms of replacement of animal models, are MPS models more cost efficient than corresponding animal studies in terms of possibility to facilitate transition to more humane drug discovery, or are they just more efficient, like more output in shorter time? So I don't know if there's anyone specifically that wants to start. If not, I can just kind of go across my screen here.
Sure, I can just start on that. Just as one example, that one study I talked about with Sanofi would have cost $2 to $3 million and take 2 to 3 years to create a humanized animal model for it. We did it in 6 months for under $500,000. Now, I'm not saying that in every point that the MPS will be cheaper or less expensive than the animal models, but certainly for most of the cost analysis we've done, it should be, especially for large animal models. Rodents may be kind of on par, certainly for mice, not necessarily for rats, but for large animals, absolutely. And you're going to get it generally faster, as well. So, that's my experience so far with this.
Yeah, thanks, James. Does anyone else have anything different they want to add?
Not necessarily much different than what Jay said. Jay covered it very well. But I think the flexibility of being able to model whatever you want in vitro is also there. For example, we have XLMTM models that there's really nothing out there. There used to be a colony of dogs available, but it no longer is available. So there's nothing. But there are kids with that disease, rare disease. So that's the other aspect of these in vitro MPS platforms that is very important aside from the cost.
Actually, Heaven makes a really good point too, in that the studies we did where we did it with cardiac and liver, with and without liver, to show the effect on metabolite. Obviously, you can't do that in animals. And you can't do it in humans either. So that's the only way would be through a humanized MPS.
Yeah, thanks, James. Anyone else want to add anything different?
I guess I could just say that in terms of cost, it a little bit depends maybe on what MPS model you're interested in using, whether it's purchasing it or whether it's contracting an MPS company to do your experiments. So there's a lot out there, and I think price is an interesting point to figure out and to compare what we can offer as MPS providers as related to the gold standard and maybe what's expected within drug development. So I think we're all working towards creating the most affordable, cost-effective options. I think oftentimes cost is also quantified by the amount of time it might take to train or onboard or become comfortable with these more advanced tools. So the more that we can all make these tools easy to use and automatable, I think that will drive down costs as well.
Yeah, great point, Lizzie. Awesome. I'm just going to go back to a question asked near the beginning, which was how to optimize co-culture media in multi-tissue modeling. So again, I think kind of a generic question for everyone. So maybe we can start with someone who hasn't spoken yet, either Ilka or Shreyas, if either of you Want to comment on that?
I can tell a bit about it. So in terms of optimizing the media for co-culturing, I would say it's a part of trial and error method. In one of the experiments when we were trying to co-culture specific, brain specific endothelial cells, we had two different sources and co-cultured with the lung tissue and the endothelial cells. And when we had to optimize the different ratios of both the medias. You have to optimize that ratio and test the viability of the cells, and the morphology of the cells is something to keep in mind. And then you will understand after using multiple different types of ratios, which is the best one working for them. And you can also test for the markers on those cell types, whether you see that specific expression which you want to see on those cells. That helps a lot.
Yeah, thanks, Therese.
Maybe adding to this, we've actually also noticed that the more organs you have, the less growth factors and cytokines and everything. Like the growth media are packed with specific growth factors, but you don't need that. As more organs you put together, they support each other more and more. We've once had like iPS-derived autologous chip on a very, very basic medium, and it didn't add any growth factors to it, and it actually got more mature over the time instead of de-differentiating again. So we actually see that we can keep them on a very simple medium because multi-organ, they support each other. That's the beauty of the multi-organs component, right?
Yeah, thanks, Ilka. Anyone else wanted to add anything? Sorry, Erica, you as well. I know you hadn't spoken yet.
Yeah, for us it's the same. Like we usually optimize it one component by adding different components, but usually the starting point is kind of mixing, really basic mixing the different medium component of the different cell types, and then moving from there.
Yeah, thanks, Erica. James, I know you were going to say something.
Yeah, I was letting everybody else to give it a chance, but as I mentioned during my talk, I had banned the use of serum in my lab 25 years ago. And so everything we do is in serum-free media, common media. We've done well over 40 cell types in basically the same serum-free media. And where the success is, people don't realize there's actually 3 variables in cell culture. Okay, it's not just the cells and the media, but it's also the surface. There's a reason why your body has 768 different integrin components in the cells. It uses those integrins, okay, to interact with the ECM to then determine, in many cases, the lineage to be able for longevity, other cell-cell interactions. But most people kind of really haven't been taught to pay much attention to the surface composition. Whereas you can tune the ECM, because that becomes your extra variable, right? Is different cell, same media, you got to have another variable, right? And so you just basically, you tune the surface for that. And that's what we've been doing for, 20, 25 years, why we have great success and lots of publications in multi-organ systems in the same serum-free media.
Yeah, thanks, James. Hamid, you had your hand up.
Yeah, the other thing I wanted to add, I think, and it's a critical question, I think the optimization of media also depends on at what developmental stage you want to bring these organs together. For example, for NMJ, do you just make your muscle tissues and your motor neurons and bring them together immediately? Do you let them mature a little bit longer and then bring them together? That changes the type of media you're feeding them. And I think that's an important question that luckily there's a lot of providers here and there's a lot of solutions, but that's a question that can be answered with one of these technologies out there or a combination of a lot of these technologies.
Yeah, thank you, Ahmed. Lizzie, I don't know if you wanted to add anything else to that question.
No, I think everyone's covered it.
Awesome.
Thanks.
Great. Okay, another question for everyone. Do you compare off-target effects in MPS multi-organ systems with effects of drugs in separate cultures of respective organs, and organs in quotes? Does anyone want to start with that one?
I think I was the only one who presented on that. So, no, they're in the same system. So, like the opioid overdose and recovery, looking at the off-target tox in the same system, there we have the targets in there, as in terms of the shutting off the prebiotic neurons or nociceptors in this case, and then turning it back on with loxone, so basically the toxicity recovery, but then also the off-target toxicity. We take that out for multiple doses. That's all done in the same system, out for 14 days. We've done some work. We published with L'Oreal where we took the systems out for 28 days. Again, all in the same media system and not done in separate ones. And again, the beauty of that is if we have a liver in it, we can see both the effect of parent and metabolite without knowing what the metabolites are, which can be very advantageous at the medicinal chemistry stage.
Yeah, I can only agree to James. I think it needs to be in a single chip, and then you can see like some of the targets. Like we had it once, but we had applied a cream on a skin, and then it had to pass through the skin first, and then only after 48 hours, you could see, and after repeated application, you could see some metabolites appearing. So it was really interesting to see that it's only working with different organ models in one chip. So you have to have the multi-organ approach. It would not work in a single organ approach.
Great.
Yeah,
Thanks.
maybe I can add on this as well, because like for us, as we have valves system, we can really take advantage of this to isolate the compartments and also elucidate what the effect of compounds and eventually metabolites on the single organ within the same device. So we are doing both, let's say.
Yeah, thank you, Arka. I'm not sure if this question was for someone specific. I can't remember, but the question is how to avoid compound absorption on MPS made of PDMS.
You can't.
Don't use it.
Well, or what we do, as I showed, is we figure out what the absorption is before we start the experiment, and you just build that into the PK/PD model. Okay, because no matter what plastic you use, even there are these low absorption plastics, if you look at them, all of them still absorb. So if you create a hydrophilic polymer, you're going to get hydrophobic interactions with it. Create a hydrophobic, okay, you're going to get hydrophilic interactions with it. You're going to get all molecules will absorb, to some degree on various polymers. There's no way to stop it. Even I saw a talk recently, somebody said they had come up with a polymer that doesn't absorb. But the last 10 compounds they showed had 50% absorption. 50% is a big number, especially when, that was acute experiments. We want to do things out to 28 days. Okay, you need to be able to understand this and predict this. And that's where the area of the curve and the in silico pairing with the in vitro is important because it's the whole idea of digital twins, right? And so you can do digital twins and pair your in vitro to predict your in vivo, but these are all variables you have to account for.
Yeah, thanks, James.
Lauren, just to add on that, sometimes, in models, you have these complete lumens, and in that case, because the barriers are so tight knit together among the endothelial cells, you have a complete lumen. The drug might not come in contact while dosing, and that's when in those situations it might be helpful, to have those models to study that if you are concerned about PDMS. If you have a complete lumen, then in most of the cases you don't have to worry about that.
Thanks, Therese. There's another question that I think is kind of interesting, concerning the shared culture media for multi-organ systems. Have any of the presenters already used AI as a first step in order to tune it properly? Anyone?
We haven't used it for developing the medium, but we're using it for analysis. So we've stained our cells without staining them, for example, or to see like how are they actually moving over the time. If we have some, for example, spheroids, we can really see if they're during the experiment already with AI, we can really see if they are growing like cancerous cells. Is it like growing a little? Is it growing much? So this is something you can really do during the experiment and analyze it directly with AI. But for the media composition, at least at TissUse, we haven't done it. I'm not sure if anyone else can add anything or has experience.
We also started using AI only in similar cases where we are trying to see how the immune cells interact with other cells, what is the movement, what is the dynamics. So that's where we started using it.
Yeah, I mean, I agree with both of them. What we use it for is data analysis. Machine learning, it just that's definitely for sure using it, but not for design. Because again, we came up with the serum-free media formulations before AI was really a thing. It just means I'm old.
Yeah, thanks everyone. It's interesting.
I mean, I'm looking at all these young faces and I see the future of MPS. This is awesome.
Yeah, great point, James. Thank you. I'm just taking a look, and I think we've answered most of them. There were a few for you, Lizzie, but it seems like you've kind of replied to those people to contact you.
Yeah, I wasn't sure if there was going to be time, so please reach out. I'm happy to answer those either live or through email.
Yeah, we do have time. I don't see any other questions in here, so I don't know if either of those questions you wanted to take a second to speak on or a few minutes.
Yeah. So it looks like one's from Tito. More about the vascular kits. So do they work through angiogenesis and vasculogenesis primarily? The angiogenesis, the angioprime product that I was referring to is angiogenetic sprouting. So the cells are seeded into a media channel adjacent to a 3D collagen hydrogel, which then allows the cells to sprout into that, undergoing angiogenesis in the presence of concentration gradients. And then the vasculogenesis assay, it's sort of like a kit that you can get. So if you wanted to vascularize your tumoroid, your biopsy, your organoid, whatever it is, we provide you with the platform and the cells and the ECM in order to be able to achieve that. So that would be a vasculogenesis process. And then there was one from MNB. So how can the spheroid be integrated into a microvascular network? Is the spheroid fabricated separately and then inserted into the device? So yeah, our platforms are intended to not grow your spheroid or your organoid, but rather you've grown that under whatever conditions, and now you're transferring that to a 3D tissue environment. Maybe you want to vascularize it, maybe you want to add immune cells. So that's where the spheroid would be added into the hydrogel that contains the mixture of cells that could then vascularize the spheroid.
Awesome. Thanks, Lizzie. I don't see any other questions, so that will wrap up the rest of the webinar for today. Just a few last reminders. Again, this webinar will be recorded, so stay tuned for that. I'll also be sharing contact information from all of the presenters with everyone, so you can reach out to them if you need to. And yeah, just a big thank you to everyone who attended. And then, of course, all of our amazing presenters today. Always really great to hear you guys speak and to see all of your interesting data. James, did you have something you wanted to add?
Yeah, I just want to reiterate Lizzie's point. The MPS Society meeting will be in Brussels, June the 9th to the 13th. If you're curious about the field, if you want to know what's going on, if you want to get the latest regulatory updates, we did it in Brussels specifically so the EMA could come down. We're going to have a large EMA presence. We're going to have also some of the legislative folks from Brussels there. We have over 1,500 registrants. We've got over 1,000 sponsors. Everybody here is going to be there. Okay, if you got more interest, you get interest in this, please come. It's not too late. Okay, registration is still open. So please, if you're interested, come join us.
Yeah, thank you so much, James. And I know we have a few of our 3Rs collaborative companies that will be there as well. So
I think all of us, right?
Yeah, amazing. Great. So I'll end the webinar there, but thanks again, everyone. And don't forget to register if you haven't yet for day 2, which will be on Thursday.